Hi, so I'm going to present our work on Kinodata 3D, which is a project on assessing whether we can use guided docking as a data generation approach for structural machine learning. And this work was done mainly by myself and Joshka in the front row right there. So he's also available to answer questions later at our poster upstairs and probably in the question session after the talk. So mainly our domain are kinases, which are a pretty well understood class of proteins. And we already saw EGFR as a case example earlier in Jessica's talk. And these are proteins that are important in many phosphorylation pathways. And especially if these pathways become dysregulated, we see these dysregulations in diseases like cancer or inflammation or other autoimmune disorders. So we have a pretty good understanding of how these proteins work. They have an ATP pocket where they bind the ATP and take the phosphate. So the aim is to find ligands that dock into that same protein pocket and block the processing of ATP for this kinase. So the general goal is to do structural models and to do these models to generate new ligands using these structural models, because the structure is what determines the function of a protein in general. But the data situation is not really that well suited to do complicated structural models, which need a lot of data to be trained. So the data we have is often in an academic setting, public repositories such as the Campbell repository, where researchers can deposit results of their binding assays. In terms of kinases, we have about 220,000 entries there, but these entries are already curated down, so we start out with a bit more, but doing some data cleaning, getting rid of duplicates, stuff like this, we end up with around 200,000 cleaned actives in this data source. But we have no idea what the structure is, how they actually bind to the given kinase. For this, we have the PDB, and the subset that is concerned with kinases is collected in the CLIFS database, which has around 6,000 crystal structures, so structures that include a ligand and a kinase that was empirically measured. And this is not quite enough to do complicated large models. So we are trying to leverage both the activity data and the structural data that we have to create a larger data set. So our main goal is to create a data set that we can use later on for machine learning. So that is what we do in this first step. We are using the Kinodata activities and the CLIFS PDB structures to do docking. And then we want to see whether this can actually do something for a machine learning task. And because we have the activity data, it's a very convenient predictive target for our models. And as models, we compare a structural model, that is the CGNN, the complex GNN on the bottom, that has access to the stock structure, the stock pose, against two baseline models that don't. So the GNN is supposed to be a baseline that is very similar to the CGNN, albeit ignoring every spatial information. And the DTI is a standard GNN bundled together with a transformer on the pockets residues. So that's the overall workflow of our study. So now let's look at the first step, the creation of the data set. So we start out with the activity data we have, which is basically just saying, here you get an activity for some given molecule and a kinase. And then we do a templating process, looking through all the available complex we have and choosing one that has a very similar ligand. So we compare the ligand in the empirical crystal structure we have with the ligand we have in our activity data. And this way we get kind of a puzzle that can be solved by template docking. So this process then takes the kinodata ligand and squeezes it into a similar pose as the template ligand was in. And this results basically in a large set of docked poses. And finally, we annotate these docked poses with a rough estimate of quality. So we're interested in how well this would represent reality. We do this via a very simple MLP that just takes diagnostic output of the docking to just annotate the quality. So in the end, we can also look at the interplay between predicted quality and filtering the data set accordingly. So if we apply this pipeline, we get out about 120,000 data points, which is a lot better than the 6,000 we had before. These mostly span PIC50 values on the right-hand side that are in the active area naturally, because people tend to more report actives than inactives. But still, we have a pretty okayish range of values in our data set. Mostly it is PIC50, so that's basically what we'll stick with for the rest of the work. In terms of predicted quality, we see a very bimodal distribution. So either we see success or failure, but we will see later how this influences the predictive quality of an affinity prediction. Yeah. And with this data, we now proceed to training the baseline models and our structural model. So let's first look at the more interesting structural model. This takes the co-complex of the ligand and the protein and has two types of bonds essentially. So we have the covalent bonds, the molecular bonds, and we have the interactions by spatial proximity. So we take an interaction radius of 5 angstrom and add edges within this radius. And the initial representation of nodes we construct out of the element, the charge, the number of hydrogens, so basic chemical features. And the edges are annotated with the atomic distance. So this is the only thing we use out of the geometric information because we want to have E3 invariance. So we don't care if we roll around the complex somehow or whatever, we transform it. We only care about the distances, and the value of the binding affinity should stay constant within that. So yeah, with this basic graph structure, we continue on with a basic message passing with attention in there and train our model based on the regression target of binding affinity. So that's the structural model we have. And along with that, we have two different baselines. So the baseline in the middle aims to be exactly that, exactly the structural models minus these dashed interactions, which are the interactions due to spatial proximity. So that's the ablative baseline, and we further have a simple DTI, which is a GNN on the ligand and a transformer on the pocket sequence. These are the 85 residues out of the kinase that correspond to the binding pocket of the ATP binding pocket. And these are then aggregated and passed through an MLP. Okay, so we use two different data splits, which are very influential on the model performance. We already saw in the peptide talk that there are lots of islands which are pretty well defined and correspond to single experiments essentially, and the same is true for Campbell data. So if you were to do a t-SNE on Campbell, you get lots of different islands in the molecular space. And basically, there's a bijective mapping between each island and one assay. So we want to see what the model would perform like in a new assay, essentially, and not what it would perform like for a very similar one. That's why we use the scaffold split. So all molecules with a similar scaffold are bundled into one part of the train test split. And similar, we split the data set according to the target. There are different kinases, and we want to see how it generalizes for completely new targets. And of course, along with both of those, we have the random split, which just, well, it's completely random. So now for the performance. We have here the test MAE, so it's the test MAE for 5-fold cross-validation. And on the very left in green, we have the DTI baseline. We have the CGNN, so the ablative baseline in orange in the middle, and in blue, the structural model. And we filter the data set according to docking quality, or the estimated docking quality we have annotated our complexes with. So if we only take the estimated good poses, we see that the MAE for the structural model is quite a lot lower than for the baseline, so it's a quite clear outperformance there. If we decrease the quality or include more supposedly or approximately bad poses, you see that it converges slightly. But even if we include these lower quality poses, the structural model outperforms the two baselines still. That is true for the random and the scaffold splitting, so the more easy cases, but for the entirely new target, basically all models are, well, a bit trashy. So we don't get a good performance out of any model for this data split. That's a lot tougher than the other ones. So if there's no data on a completely novel kinase, it's much more difficult than looking at other scaffolds. Yeah, so basically we see that structural information always helps, or if it doesn't help by a lot, it at least doesn't hurt to include even bad structural information, in this case at least. So this is a nice learning. Also, we could just say for our structural generative models, we are only using the supposedly good data. So now we are asking ourselves what else to do with this data set. And some colleagues are already doing more traditional MD simulations and stuff like that with this data set. Or interaction analysis could be done. But the models we used in this study could also be put to use in stuff like automated cut pipelines. Yes. Or the data set overall could serve as a benchmark for affinity prediction, for example. Yeah. And finally, we're excited to apply this to generative tasks, especially in a fragment-based setting, which was the original genesis of this whole project, to generate a larger data set for this. Yes. So that is the Volkamer lab on the left-hand side, which is the new group that just started about a year ago at Saarland University, and Joschka and Verena, who collaborated on this project. Yes. So with this, I thank you for your attention.
Thank you, Mikael.